Hi ladies, this is a presentation on DNA and DNA replication. You can find this information in chapter 13. So the first thing I'm going to briefly review is the search for genetic material. So initially, Morgan, who did his research on the fruit flies, he was the one who first discovered sex-linked inheritance. He discovered that there are two components of chromosomes, DNA and protein. And initially back then, nobody really knew exactly what the genetic material was. So it could have been DNA, it could have been protein. So researchers were looking for if they could figure out which one was the correct form of genetic material. So the first experiment was Griffith. If you remember, he discovered that transforming factor. So the smooth strain the rough strain, was transformed into that smooth strain. Avery identified the transforming substance as DNA. This is the experiment that extended Griffith's experiment, and he used an enzyme that destroyed DNA and found that there was no transforming factor when the DNA was destroyed. Alfred Hershey and Martha Chase, they discovered that DNA was also the genetic material, and they used bacteriophages in the famous blender experiment. Erwin Chargaff, he discovered that base composition of DNA varies between species, and he always found a similar pattern, that there was always an equal number of A's, adenines, and T's, thymine, and there was also an equal number of guanine and cytosine in every DNA sample. He wasn't exactly sure what that meant, but he did discover that pattern. From here, scientists continued to look into DNA further. Rosalind Franklin produced a picture of the DNA molecule using X-ray crystallography. You can see her image on the right. This was what first gave Watson and Crick the idea that DNA was a double helix. So they got this idea from her image. James Watson and Francis Crick also discovered additional aspects of the structure of DNA. So they determined how the nucleotide, the phosphate, nitrogenous base, and a sugar were arranged. They found that all DNA molecules were a double helix. They were anti-parallel, meaning that one side went 5' to 3' and the other side went the opposite direction, 5' to 3'. They found that adenine was hydrogen bonded to thymine and cytosine was hydrogen bonded to guanine. They also discovered that there was a phosphate and sugar backbone, so deoxyribose in the DNA and ribose in RNA. And this phosphate and sugar backbone supported the nitrogenous bases that were on the interior of the molecule. So let's look at a couple pictures of this. So you can see here all the different nitrogenous bases in the sugar phosphate backbone. And you can see what 5' to 3' truly means here. If you look, when we say 5', we're actually referring to the 5' carbon of the deoxyribose sugar. So you can see 5' carbon here. And then the 3' end, okay, it ends with the 3' carbon of the deoxyribose sugar. Okay, so this shows the phosphate sugar backbone and the nitrogenous bases on the interior of the molecule. Here you can see the double helix present in the DNA molecule. It kind of has that nice twisted ladder. Here's the hydrogen bonding, thymine to adenine, guanine to cytosine. Okay. Hydrogen bonds are perfect for DNA. It allows easy replication and transcription. Later, we will talk about that later. So it's a really weak bond, so it can be opened up and then reclosed, but it still does provide some stability. Okay, so you can see here the structure of how adenine and thymine bond together and guanine and cytosine bond together. Adenine and thymine always form 2 hydrogen bonds. So 1, 2. And guanine and cytosine always form 3 hydrogen bond. 1, 2, 3. So that's why you can't have adenine bonding to cytosine or something like that. It just wouldn't work. The bonds wouldn't line up. What was also interesting is that the way that the molecules, the nitrogenous bases are arranged, there's always a purine bonded to a pyrimidine. Okay, so a purine is a double ring structure in the nitrogenous base, and a pyrimidine only has a single ring structure. So this is really important to produce that double helix structure, and it made it very consistent with the X-ray data. Oops. So you'll note up here, okay, we have purine is adenine, guanine is also a purine, and a pyrimidine is thymine, and cytosine is also a pyrimidine. The next really important discovery was how DNA can possibly replicate. So Watson and Crick suggested a semi-conservative method, and they thought that because there were hydrogen bonds holding together the nitrogenous bases, those hydrogen bonds could easily be separated. And then those parental strands could be used for the new strands. Okay, so because DNA is always complementary, you can use the old DNA as a template to make new DNA. This was later proved in Messelson and Franklin Stahl's experiment. So they were able to demonstrate that the semi-conservative method is correct. So what they did is they had bacteria cultured in a medium with a heavy isotope of nitrogen and bacteria cultured in a light isotope of nitrogen. When they initially did the experiment, they started with the heavy isotope and then grew the bacteria in the lighter isotope. When they did this, they found after one round of replication that the DNA was this hybrid. So it wasn't heavy, it wasn't light, it was somewhere in between whenever they centrifuged the sample. So because this happened, they can eliminate the conservative method, stating that the new copy is made from the old copy and is identical, but it's separate. After an additional round of replication, okay, in using the light medium, they found that there was a hybrid and there was a less dense copy. So this verifies that it wouldn't be a dispersive method either. So using pieces of the DNA molecule and kind of piecing them together. If this was the case, you have all hybrid. Because they found a hybrid band and they found a light band, they can tell that one of the heavy bands was used as a template, one of the light bands was used as a template, so that would produce a light nitrogen isotope strand of DNA and then a hybrid strand, and the same thing for this other strand of DNA. So really interesting that they were able to prove this is how DNA replicates. So for the last part of today, I'm going to actually go through the technical nitty-gritty way that DNA does replicate. So here's a list of terms and things that you need to be comfortable with. I'm not going to go through them right now, but I'm going to talk about them in relation to a couple of different images that you're going to see. So the first one compares the replication of E. coli and the replication, so like a prokaryotic cell to the replication of a eukaryotic cell. So the first thing you should see here is replication bubbles. In a prokaryotic cell, there is just one chromosome that's circular, and a replication bubble will start at one part of the chromosome, and it'll work its way. So you can see here's the replication fork on each side. The replication fork will work its way all the way around until you end up with two daughter chromosomes. In eukaryotic cells, there's a lot more DNA than there is in prokaryotic cells. So these cells have to form multiple replication bubbles along the DNA strand. So in many, many locations, these bubbles will form. Okay, so at an origin of replication, and then these replication forks will keep moving until they actually meet up with each other. Okay, and then those bubbles will fuse until you actually have two strands of daughter DNA. So it's a little bit different. I'm going to show you a couple of the enzymes and things that are involved. So just a couple of enzymes that I'm going to mention: helicase, topoisomerase, primase, DNA polymerase, DNA ligase, and then I'm going to talk about the leading and the lagging strand. I'm going to explain this with an image, but I do recommend that you go back and review and look at the videos that I've also posted. So here is an overview of what's going on here. So you have one big picture. This is one of those replication bubbles. Okay, origin of replication, is where DNA replication is going to start, and then the bubble is going to expand to the left side and to the right side. So I'm going to look at the leading and the lagging strand for a second. The leading strand is a continuous strand of replication. It moves towards the replication fork right here, and it can be copied continuously in one piece. The lagging strand has to be copied in multiple pieces. Okay, the reason this happens is because DNA is always created in a 5', the new nucleotides are added in a 5' to 3' direction, always. And that's really, really important. So how does this work? You have topoisomerase, which is actually going to help stabilize the DNA and kind of almost untwist it a little bit. So that way it's more flat and stable. So that way because normally it's a double helix and it's kind of like in that twisted ladder. So topoisomerase is going to untwist and kind of stabilize the DNA. And then you have this enzyme right here at the replication fork, helicase. Helicase will always be at the replication fork, and basically it breaks the hydrogen bonds that hold the parental strands together. So here is helicase. In order to add a new DNA fragment, primase has to come on, and primase will add an RNA primer. It's only about 5 to 10 nucleotides of RNA, and it's basically like a start here sign for DNA polymerase. DNA polymerase will find one of these primers put down by primase, and it will start adding DNA nucleotides in the 5' to 3' direction. Okay, so you can see DNA polymerase here adding new nucleotides. On the leading strand, DNA polymerase is going to add nucleotides continuously, okay, towards the replication fork. It never needs to stop. But on the lagging strand, it's going to have to add them in pieces, okay? Because it goes 5' to 3', it's first going to add a piece, okay? And then helicase is going to unwind more of the DNA. So then you'll have another primase come in here, put a primer down, and then DNA polymerase will follow and add another piece to where this primer is, okay? As helicase keeps working, it's going to make more space for more DNA to be added. So this has to be done in pieces just because it has to be added in the 5' to 3' direction. All of these pieces are what we call Okazaki fragments. And once this is complete, what actually has to happen is DNA ligase right here has to come and put those little fragments together. So it's going to fuse those Okazaki fragments into one continuous piece. So that is basically how DNA is replicated. I recommend that you watch the videos because they can show a little bit better how this works, but we'll talk about this a little bit more next time we meet. Just one final thing to talk about is the evolutionary significance of altered DNA nucleotides. So sometimes when DNA polymerase is adding nucleotides, it makes a little mistake. This is very rare, but it's not completely unheard of. So the error rate is very low, but it's not zero. And when these changes happen, these are what we call mutations. So they're a source of genetic variation, and these mutations are really, really important. They can change a gene and cause a new phenotype to be created. This new phenotype may have an evolutionary advantage, so it'll be able to survive and reproduce more than other individuals. So sometimes these mutations are good, sometimes they could actually be bad, and that would usually be eliminated in the population, but there is the possibility of having good mutations or bad mutations. So that is DNA replication, and I recommend that you watch both replication videos on Haiku. Please bring any questions you have with you to class.